So one of the things we need to understand about chromosomes is that half of the genetics in our chromosomes are going to come from our mom, and the other half are going to come from our dad. So we're going to call the ones that come from mom maternal. And we're going to use this Lego as our center point on our genes for the maternal side of the chromosome. And this is called a centromere. And for the paternal side, we're going to use the black Lego. Now, to represent the different types of genes that could be present, I have different colors to represent different traits. We're going to be looking at placement of traits and not so much as like this one is specifically eye color, right? But that will be helpful in understanding later on how genetic differences occur. So let's take a look at a full-size chromosome. Now remember, this is just an example. It's not perfect. All right, this is going to represent chromosome 1 for my fake species. On this first set where the gene codes, it's going to code for eye color. Remember, each little individual Lego represents a gene. So in this chromosome, actually, it codes for 1, 2, 3, 4, 5, 6 genes. This one's eye color, blood type, skin color, hair color, amylase, and hemophilia. Some of those words might sound familiar, like amylase. That's an enzyme that breaks down carbohydrates in our mouth. Hemophilia, that's a blood disease where you can't stop bleeding. No hemophilia versus hemophilia. So our genetics are encompassed in our chromosomes. Everything from diseases to the enzymes we're making to the color of our hair and our height and who we are, right, is on the chromosome. So let's look at the structure of the chromosome for a second. On this side, I have my maternal chromosome, remember? Cream color. And on this side, I have the paternal chromosome. So these genes came from the dad, and these genes came from the mom. This is going to become really important later on, because these genes from mom and dad are a mix-up of your mom and dad. And we're going to show how those mix-ups become gametes. We're going to go ahead and stick to our original chromosome and talk a little bit about replicating this strand or these two strands of DNA. So remember that when you take the chromosome apart, you now have two chromosomes. When we go to replicate, whether we're replicating for the process of mitosis or if we're replicating for the process of meiosis, we have to make a copy of this. And if you remember, that's DNA replication, and it's way more complicated than this, right? But ultimately what you're going to do is you're going to make an exact copy of that strand of that chromosome, or that chromatid, of that chromatid. So here is my example. I have my two sides of my chromatid, the one from dad and the one from mom, and I've made an exact copy of them. Now, to make a whole functioning cell, if you remember, in mitosis, right, you need to have a copy of each of these genes. So they line up in the middle, and then they will be pulled apart to the opposite side, where then they become two new cells with that chromosome, that original chromosome, present. So there's my example of that process. Now remember, that's mitosis. All right, let's start diving into meiosis. So chromosome 1 is right here. Remembering that we have our genetic markers here for eye color, blood type, skin color. We'll come back and kind of look at the end result of one of our gametes and see how that has changed. On the paternal side, we have all green, and on the maternal side, we have all red. Now we'll see how that changes once we go through our first steps, the first stage of meiosis, which is meiosis 1. All right, so I'm actually going to add in another chromosome. I'm going to add in chromosome number 2. We don't know anything about chromosome number 2, but we're going to use it to help us understand. So chromosome 1, chromosome 2. The first step that we need to do, as the same step in mitosis, is replicate the chromosome. To do that, we take the chromatids, so we take the chromosome apart, and we have our chromatids. We're going to replicate those chromatids. Now, I know it's a very simplistic idea of replicating my chromatids, but here we are doing the replication process. And you'll notice that they should be identical to the one that they are attached to. And they are. They're identical at this point. I've got my chromosome 1 and my chromosome 2. The paternal side are the black ones. The maternal are the cream color. And we just replicated that strand of DNA. So if I put this on here, back into our paper, this is the same side as that. All right. There are two things that are going to occur to help get genetic diversity, which is extremely important, and it's why you look different than your brothers or sisters, if you have them, and why you also look different than your mom and your dad. The first one is called crossing over. Now, crossing over is going to happen between your two sister chromosomes here. All right? And crossing over is going to happen at specific genes. And it's random. It's very random. So, I'm just going to randomly switch around some of these genes. So, keeping them in the same position, I'm going to take this chunk, and I'm going to switch it with this chunk. That's a big change. All right? Let's do a smaller change. I just want to change this individual gene. So, I'm going to take out the gene. I'm going to swap it with the gene that's in the same spot. Okay? So what I've done here is I've taken the 2 copies of my maternal side and the 2 copies of my paternal side, and I've switched them up a little bit, creating some genetic diversity. I'm going to go ahead and do that here with my smaller, but my second chromosome. And remember, it's all very random in the process. Oh, I put that on backwards. I thought so, because what switched out was this gene. All right, so we have a variety of different genes that have been switched out. That process right there is called crossing over, and it happens on all of the genes from your body, on all of the chromosomes that are in your body. Remember, it's happening between the maternal and paternal side of the chromosome. Not chromosomes to chromosomes, right? But from the maternal to paternal side. Okay? Now, the other process that helps with creating genetic diversity is going to be independent assortment. And really what that means is it's a fancy term for how the genes line up in reference to their original maternal and paternal chromosomes. So for instance, I've switched on this side is my maternals and on this side are my paternals. That's a pretty good genetic diversity. I could keep it the same. Paternals, maternals. You add more genes, they can mix up and end up in any position. But they will line up next to each other based on their chromosomes. So these are called sister chromosomes. All right. Now we're at the process where meiosis is going to start creating two new cells. And it needs one of each chromosome, one paternal and one maternal in the new. So it's going to, not yet. It's going to take this side and it's going to divide it over here. And this side is going to come over here, and these are going to be two new cells. All right, guys, so we've got our genetic diversity here that we've created. We did some crossing over between our maternal and paternal chromosomes, and then we did some independent assortment here. We're noticing that I have a maternal and a paternal here, and then a paternal and a maternal. Now, before we move forward, I think it's important to look at how the crossing over process provided some genetic diversity. So let's go ahead and re-look at our map of our chromosome 1. So this is our paternal and this was our maternal. If you remember correctly, the paternal was all green and the maternal was all red. So you'll notice where the changes are, because in the maternal, wherever there's a green, that's a change to that original strand of DNA. So there's a change here in blood type, and there's a change here in hair color, amylase, and no hemophilia. Okay? Remember, that was all maternal. When we look at our paternal, okay, you're going to notice those are the same spots that have changed. But now what should all be green, we now have these new red ones that have been placed in here. So we're getting a variety of genetic diversity. So here we are. We're all lined up. We're ready to actually start our meiosis process. We're actually in the middle of it already for meiosis 1, but we have now gotten into our metaphase of meiosis 1. So metaphase 1. In metaphase 1, these sister chromosomes are going to line up, and the spindle fibers are going to come in. They're going to connect to our centromeres, and they're going to pull these apart. So, replicating the pulling apart process, which is anaphase, you will notice that over here I have the original maternal and the original paternal for chromosome 2. And here I have the original paternal for chromosome 1 and the original paternal or maternal for chromosome 2. These become two new cells that will then divide in our next stage of meiosis, which is meiosis. Now this is where we're going to see some big changes. In meiosis 2, remember meiosis 1, we have the two sets, but we're going to actually divide this set, and then we're also going to divide this set. So let's start with one set. Move this one out of the way so we can show our process here. So we're going to be doing metaphase 2. So in metaphase 2, the chromosomes are lined up, and now we're going to split them into their chromatids. So now we're in meiosis 2 anaphase. And you'll notice now, okay, that what we have are haploid cells, and they only have half of the genetic material, and they will become their own cells. Whether they are an egg cell or a sperm cell, just depends. Well, doesn't really depend. It's all based off of the genetic material and whether it's a male or female. Then the other cell will go through the same process, and they will line up for meiosis metaphase 2, and then meiosis anaphase 2, and they will split. Now we have more genetic diversity. So now I have my cells. Cell 1, cell 2, cell 3, and cell 4. You can see that they're all very different from each other and have a variety of genetic information. When they combine with their opposite gamete, they will then come together and form a new cell that has the full amount of chromosomes, which will be the other half of this specific chromosome.